I'm very sorry that I cannot attend in person.
Oh, yeah, but next time for sure you will be here. We know. So.
Yeah, yes, I will. I will. Okay.
Perfect. So, without further ado, we will hand over to you, Kozo, and enlighten us. Thank you very much.
Okay. Can I start?
Yes, you can start. Please, go on.
Okay. Yeah. So thank you for the introduction. Good morning, everyone. So I'm sorry that I cannot attend in person, but I'm delighted that the second CHAMP1 conference in Italy is going so well. So the first talk in today's session, I will introduce an overview of the CHAMP1 research. I hope this will help you understand the subsequent talks. Okay. All right. Okay. Merit has sent me a list of questions she will like me to address to the audience in my presentation. As this list is quite comprehensive, so that I will give my talk according to it today. The first question is on the path from gene to protein. So we have about 90,000 genes in every single cell in our body, which are on our genome, which is all the genetic information written in the form of DNA sequence. And this genomic DNA is folded into chromosomes. Our genome is made up of 3.1 billion base pair of DNA, which exist in the form of 23 pairs of chromosomes, 46 in total. Our 90,000 genes are distributed across these chromosomes, and the CHAMP1 gene is located at the end of chromosome 13. A gene is a blueprint of a protein, and the information is first copied to RNA, which is called transcription. Then a protein is synthesized by adding amino acids according to the information in messenger RNA, which is called translation. As this pathway from DNA to protein is conserved across species, it is called central dogma. When messenger RNA is used as a blueprint for a protein, a combination of 3 nucleotides, shown as UCAG, correspond to 1 amino acid as shown, which is called the genetic code or codon. Therefore, gene mutations lead to the formation of altered proteins by incorporating different amino acids. The second question is on the types of mutations. Types of mutation can be divided into 2. Mutation at the chromosome level and those at the DNA level. While large genomic regions containing multiple genes are affected in the former, single genes are affected in the latter through alteration in short DNA sequences. These are the mutations at the chromosomal level. You may have an idea from their names and these schematics. They can be recognized at the microscopic level and often found in cancer. On the other hand, these are mutations at the DNA level. If a corresponding amino acid change to another amino acid by the mutation, it is called missense mutation. If the corresponding amino acid is changed to stop codons, which stop the elongation of amino acid, it is called a nonsense mutation. On the other hand, when some nucleotide are lost or gained, it causes a shift in the leading frames, which is called a frame shift mutation. When it occurred, the amino acid sequence are totally changed thereafter, and the elongation is eventually stopped by the appearance of stop codons. Therefore, nonsense or frame shift mutation give rise to truncated proteins. I will also explain heterozygous and homozygous mutations. As we are inheriting the same set of genes from father and the mother, we have two copies of a gene, and proteins are made from both copies. When one of the copies is mutated, it is called heterozygous mutation. In this situation, normal protein made from one copy and mutant protein is made from another copy, making the amount of the wild-type protein halved. The function of most genes are generally maintained even when one of them is mutated. However, the function of some genes are affected by this mutation, which is called haploinsufficiency. When both copies are mutated, it is called homozygous mutation. In this situation, only mutant protein is made, which is seen in cancer cells and also seen in some rare genetic diseases. Next question is on the outcome of mutant proteins. This is a diagram of the CHAMP1 protein. And since 2015, CHAMP1 mutations in individuals with neurodevelopmental disorders have been reported in many papers as shown here. Three types of mutations are missense mutation, nonsense mutation, and frame shift mutations are shown in blue, red, and green respectively. As you can see, most of the mutations are nonsense or frame shift mutations, and they are de novo mutations, which means they are not inherited. And in some cases, the whole CHAMP1 gene is lost by deletion, and some of them are inherited in these occasions. However, the CHAMP1 Research Foundation has much more information than these papers, and as shown here, now it is found that there are many cases of missense mutations throughout the CHAMP1 sequences, as shown in blue. And some of them are inherited. As I explained, nonsense or frame shift mutation in CHAMP1 give rise to premature termination codon mutants, as shown here. We and Dr. Alan D. Andrea groups confirmed that truncated CHAMP1 proteins are expressed in cells from CHAMP1 children. These bands correspond to these CHAMP1 mutants which migrated at expected sizes. What are the outcomes of CHAMP1 mutations? As the mutant CHAMP1 are expressed in cell, as the mutant CHAMP1 lacks a part of a protein, it may not function as wild-type CHAMP1, In this case, only a half amount of wild-type CHAMP1 function, which is called haploinsufficient. The second possibility is that the mutant CHAMP1 perturbed the function of wild-type CHAMP1, which is called dominant negative. I will explain this later. The fourth question is on the function of CHAMP1. So our group have found that CHAMP1 has several functions, such as maintenance of kinetical microchip attachment and double-strand break repair and cell survival during cell cycle arrest and neuronal development. We first define CHAMP1 in its role in chromosome segregation. Chromosome segregation is a process during cell division in which previously duplicated chromosomes are segregated into two daughter cells, as shown in this field. This process takes place on the spindle, which is made up of molecular fibers called microtubules. And for chromosome to segregate properly, they must first align themselves with the center of the spindle. These panels show chromosome alignment in normal cells. In contrast, in lower panel, the expression for CHAMP1 has been reduced. As you can see, the chromosomes are misaligned due to insufficient attachment to microtubules, resulting in chromosome segregation defects. We found that the middle region of CHAMP1, which contains a lipid motif called the FP motif, shown in green, is required for the role of CHAMP1 in chromosome segregation. We also found that CHAMP1 is highly phosphorylated in mitosis, which is required for its role in chromosome segregation. These results led us to name this protein CHAMP1, short for chromosome alignment maintaining phosphoprotein. We found that CHAMP1 is also involved in DNA repair. DNA in our bodies is constantly damaged by various insults, but we survive thanks to various mechanisms that repair these damages. Among DNA damage, DNA double-strand break, in which both strands of the DNA double helix are broken, are the most serious damage, and the cells cannot survive if they are not repaired. This graph shows the survival of cells exposed to ionizing radiation that causes DNA double-strand breaks. Even normal cells, in blue, die as the dose of radiation increases. When the expression of CHAMP1 was reduced, the cells died to a greater extent than control cells, showing that CHAMP1 is required to repair DNA double-strand breaks. There are two pathways to repair DNA double-strand breaks. One is non-homologous end joining, which is like sticking broken sticks together with glue. In this process, broken pieces of DNA are often lost. Therefore, this repair is not accurate. However, when genomic DNA is doubled to prepare for cell division, cells can use their intact counterpart of the DNA strand as a master copy to accurately copy the lost information for repairing DNA double-strand breaks. This pathway is called homologous recombination. And we found that CHAMP1, together with POG-Z, promote this pathway over the imprecise non-homologous end joining pathway. The role of CHAMP1 and POG-Z in DNA double-strand breaks repair is also shown by Dr. Alan D. Andrea's group. Recently, Alan's group investigated the role of CHAMP1 in this regard further and reported that CHAMP1 complex promotes heterochromatin assembly to maintain a genomic structure. We also reported the analysis of CHAMP1 knockout mice, in which one or both copies of CHAMP1 genes were deleted, while homozygous knockout mice could not survive. Heterozygous knockout mice exhibited characteristics similar to those of CHAMP1 individuals, such as mild memory defect and altered social interaction. This is the cerebral cortex of a newborn mouse. Neurons are shown in green, and in the developing brain, young cells move to the top as they become mature neurons. Here, you can see some green cells moving up. After 2 days, which is 1 day before birth, many cells have migrated up and formed the layer structure. In contrast, when CHAMP1 expression was reduced, you can see here and here that the number of cells rising to the top is reduced, showing that the migration and the development of neuron is delayed. These data suggest that CHAMP1 knockdown leads to a delay in neuronal development. Okay, the next question is on the molecular structure of CHAMP1. This is a schematic of CHAMP1 proteins. CHAMP1 is 812 amino acids long. Proteins can be divided into domains that have different structures and functions. CHAMP1 has zinc finger domains at both ends, shown in blue. In the middle region, there are several types of repeat sequences shown in different colors, and this region does not form a discrete structure known as the intrinsically disordered region. Zinc finger domain is a structural protein motif that uses a zinc ion to stabilize its structure, and like here. So these domains are often found in eukaryotic transcription factors and are crucial for binding to DNA, RNA, and proteins. On the other hand, intrinsically disordered region is a segment of a protein that does not adopt a stable, fixed three-dimensional structure. Despite their lack of a fixed structure, they are critically important for many biological processes. From this structure, it was predicted that CHAMP1 has globular domains at both ends connected by a flexible region. This was confirmed by this movie, which was taken using high-speed atomic force microscopy. You can see globular domains at both ends connected by a flexible region. Please note that the CHAMP1 structure may be stabilized inside cells by binding to other proteins such as POGZ and to DNA. Next question is on the complex of CHAMP1 with other proteins. This schematic illustrates the molecules interacting with CHAMP1. 3 proteins have been characterized as CHAMP1 interacting molecules: POGZ, LEB7, and HP1. Among them, POGZ is also frequently mutated in neurodevelopmental disorders as shown here. This is a diagram of a POG-Z, which also has zinc finger domains shown in yellow and bind to CHAMP1 at its tail region. Here, the mutations are colored according to whether they are seen in autism spectrum disorder, intellectual disability, or other neurodevelopmental disorders, collectively known as White-Sutton syndrome. Interestingly, POG-Z was originally reported for its role in proper chromosome segregation, like CHAMP1. And POG-Z is also involved in homologous recombination gene repair, in addition to neuronal development. These overlapping roles of CHAMP and POG-Z highlight the close relationship between them. A number of papers have been published on the analysis of POG-Z knockout mice, or mice with POG-Z mutation, which are informative in considering the role of CHAMP1. Many of these papers suggest that POG-Z plays a role in transcriptional regulation that may be associated with neurodevelopmental disorder. Among them, Irene's group at the Cornell CARS suggested that POG-Z, together with HP1 and ADNP, another causative gene for neurodevelopmental disorder, played a role in transcriptional regulation in a context-dependent manner. So sometimes they suppress their gene expression, and then in some cases increase gene expression, which is reduced in POG-Z knockout. When a gene is transcribed, many proteins, including RNA polymerase II, which is crucial for gene transcription, work together by binding to an upstream of region of DNA called the promoter. In addition, a distal region of DNA called the enhancer is also involved in transcription regulation, as shown here. One possibility is that CHAMP1 works together with POG-Z in this process. These images show the reduction of neuronal gene expression in embryonic brain of POG-Z knockout mice. So these are name of genes which is related to neuronal development. And compared to the left, which is a wild type mice, so the expression of these genes are shown in blue, are reduced in POG-Z knockout mice. Interestingly, we found that the expression of most of these genes were also reduced in CHAMP1 knockout mice, as shown here. So these, so the marked genes are also reduced in CHAMP1 knockout mice, suggesting that CHAMP1 and POG-Z work together on the transcription of these genes. Genomic DNA is wrapped around histone to form a structure called chromatin. So when chromatin is tightly wrapped, as shown here, transcription factors cannot access the DNA, and thus transcription does not occur. To start transcription, chromatin DNA need to be regionally free of histone to allow the binding of transcription factors, which is called open chromatin. So group of proteins called chromatin remodeling complex plays a role in to regionally open this chromatin structure. We think CHAMP and POGZ play the role in this process. To examine this possibility, we are working together with Stefano on genomic localization of CHAMP1 in mouse embryonic brain. This is an example of the CHAMP1 localization on a gene, which is shown as peaks on genomic regions. So as you can see, CHAMP1 localizes around the transcription start site. So that this is a genome sequence of a gene, and this is the transcription start site. So these regions are promoter regions. So okay, so the which is typical. So the localization to the this promoter region is typically seen in transcription regulators. So as you see, POGZ is also on the same genomic region, and we found that more than half of the POGZ peaks on genomic region overlap with the CHAMP1 peaks, suggesting that, as shown here, CHAMP1 and POGZ cooperatively regulate expression of neuronal genes, so that which we are now working further with Stefan. Okay, and the last question is, what is still unknown about CHAMP1? So this is a good question. So these are the list of the major genes associated with neurodevelopmental disorders. Other genes, so the genes other than that are also involved, but these are major genes. But still, there are 93 genes. And then POGZ and CHAMP1 are included in this list. Okay. So these are the number of papers reported so far on these genes. So there are, this means the number of scientific paper studying on these genes. So far, 35 papers were published on CHAMP1, and many of them are case reports. And on POGZ, 103 papers were published, which is more than CHAMP1. I apologize, I did not have time to check all other genes. But as you see, more papers, sometimes more than 10,000 papers were published for other genes. So in this sense, CHAMP1 research is only just beginning compared to other causative genes, especially on the mechanism of neurodevelopmental disorder. However, whatever the mechanisms, we know that they are originated from CHAMP1 mutations. Therefore, whether CHAMP1 mutants behave as haploinsufficient or dominant negative is an important question. Recently, we have addressed this question in the role of CHAMP1 on homologous recombination. Although I don't show data today, we found that in our experimental setting, the CHAMP1 mutants behave as haploinsufficient. In contrast, Marcella's group and Dr. Baxbaum's group reported that these mutants behave as dominant negative regarding the severity of the symptom of neurodevelopmental disorders. So there is a discrepancy. So these data suggest that deficiency in homologous recombination may not account for the cause of neurodevelopmental disorders. And transcription regulation may be related to neurodevelopmental disorders. In terms of transcription regulation, in the case of haploinsufficient, a half amount of wild-type CHAMP1 is not sufficient to carry out proper functions. On the other hand, in the case of dominant negative, mutant CHAMP1 competes with wild-type CHAMP1 either by binding to POGZ or binding to DNA, kicking out wild-type CHAMP1. So this is the last slide. One of the potential therapeutic approaches is to increase the expression of wild-type CHAMP1, so which will improve the situation in the case of haploinsufficient. So here, now the amount of CHAMP1 is sufficient to carry out the proper function. Probably in the case of dominant negative as well, so that if the level of wild-type CHAMP1 increases, so that this wild-type CHAMP1 has more chance to and then win the competition between mutant CHAMP1. Okay. Several researchers in CHAMP1 Research Foundation are now working in that direction. Finally, I would like to thank all the collaborators in CHAMP1 study, including Stefano. And then, and I would also like to thank the CHAMP1 Research Foundation for the continuous support for our study and for organizing such a strong research community. Thank you very much for your attention, and I'm happy to take any questions.
Thank you very much, Dr. Tanaka, for your explanation. Is there any question? Okay, let me get the microphone.
Hey, Kozo. This is Zach. Can you hear me okay?
Yeah. Hello.
Good to see you, albeit virtually. So you're suggesting a few different roles for CHAMP, a few different mechanisms of action. I'm just sort of curious if you think that there's any chance, some of the CHAMP mutants could be haploinsufficient as it relates to certain functions and possibly dominant negative as it relates to others, and why or why not?
I don't have a clear answer for that. Yeah, so that's one possibility. So yeah, so from our recent data, so that I think we have to carefully evaluate the function of CHAMP1 in different contexts. Thank you very much.
Thanks.
Thank you. Is there any other question? Don't be shy. No, no.
Hi.
Hi.
Can you explain how you enhance the wild type of the chromosome? Because when you have it in the DNA, it still has. And do you use the marrow, the bone marrow, or do you use only the mutant just in the brain?
Okay, so basically, all the cells in the body, whether bone marrow or brain, so that all the cells have the same set of genes. Although the pattern of gene expression differs between cell to cell, so neurons and blood cells, but still the genomic information is the same. So the only the expression is regulated depending cell to cell. So yeah, if you examine the genome in one cell, you can reliably say this is also the same for all other cells.
Okay.
Yeah. Okay.
Yeah, but when you will enhance it, it has to be from the stem cells, isn't it? Because you will it changed.
Stem cells. Yeah, even for stem cells, genomic sequencing itself is the same. Am I answering correctly to your question?
Yeah, you're correct. But as the person is done, but when you will change, you have to be the stem cells to be developed.
Okay. So are you talking about the mouse experiment?
Yes, yes.
Oh, okay, yeah. Oh, that's important. That's important. Yes. So we cannot change, the genomic information for individual cells. But in the case of knockout mice, we change the genomic information in embryonic stem cells. So then the newborn mice. So that originating from this mutated embryonic stem cells will have this altered genetic information for all the cells in the body. Okay.
Okay. Yeah, that's what I meant. Thank
Okay.
you.
Thank you.
Okay. Are there other questions? No? Well, thank Dr. Tanaka for your presence here. It has been a
Thank you very
pleasure
much.
to have you. Let's hope next time we organize in Europe a conference, you will be able to join. We will be so
Thank you, I
nice.
will. Yeah.
Have a
Thank
good
you very much.
night, because I guess that it's a quiet night for you.
Yeah.
Thank you.
Bye.
Goodbye.